Hello, everybody, and good day. It's a pleasure to be here, and you're welcome to the writing workshop being delivered by the African Journal for Laboratory Medicine. The African Journal for Laboratory Medicine is the official journal of the African Society for Laboratory Medicine, and like the Society, we're 10 this year. It's a pleasure to have you for this workshop. My name is Iruka Okeke. I'm the editor-in-chief, and we have a great lineup of speakers to share some of their insights on manuscript writing today. For those of you who applied and were accepted into the workshop, there will also be an interactive session on Slack. So to get us started, I'm going to introduce you to our journal's managing editor, Bethany Rammer. Bethany will be walking us through some of the housekeeping rules for the workshop. Bethany.
Thank you very much, Iruka, and good morning, good afternoon, and good evening, everyone, and welcome to the 5th AJLM writing workshop to take place as part of an ASLM conference. We're really glad to have you here. Again, my name is Bethany Rammer, and I'm the managing editor for AJLM, which is ASLM's peer-reviewed scientific journal. It falls under our data and communication strategic pillar. So ASLM sponsors these workshops as an investment in the future advancement of laboratory medicine in Africa. And this is a very important thing to do, as Dr. Tom Frieden said in his keynote address yesterday. Research conducted in resource-limited settings, like many countries in Africa, is too often conducted by people who don't reside in those countries, who then go on to publish without including local authors. So one of the ways that we can help end that practice is by training researchers to write better so that they can be the ones doing the research, the writing, and the publishing. So that's the big picture on why we're here today. There's also, we're also today specifically aiming to train less experienced and early career researchers. So the presentations that you'll see will be very focused on fundamental basics and mechanics of writing and submitting manuscripts for publication. So as Iruka said, the workshop is actually going to take place on 2 platforms. We'll have the presentations and moderated Q&A style discussion right here on this platform. And that is open to everyone. You don't have to have applied. So for during some of the presentations, there'll be short exercises, and those will be conducted on Slack. And the folks there's about 25 people who were selected to participate in that, and they pre-applied and were selected for the mentorship portion of the workshop, specifically because they were thought to be, they would benefit from direct writing-oriented mentorship from our workshop's resource people, which includes our presenters, some of our AJLM editors, and some members of our editorial staff. So with that, it is my pleasure to introduce Professor Iruka Okeke. She is AJLM's current editor-in-chief, and she is a member of the Council of Scientific Editors. She holds the position of professor at the University of Ibadan in Nigeria, and she is a fellow of the Nigerian and African Academies of Science. Her research group investigates the mechanisms that bacteria use to colonize humans, cause disease, and gain drug resistance. She also studies laboratory practice and has authored a number of scientific articles, as well as the books Divining Without Seeds, The Case for Strengthening Laboratory Medicine in Africa, and Genetics, Genes, Genomes, and Evolution. So once again, welcome everyone. I hope you get a lot out of the workshop, and over to you, Iruka.
Hello, everybody. Thank you very much, Bethany. So I'm just going to introduce the other speakers that we have with us today. We have Jenny Punt, who's an associate dean and One Health professor of immunology at the University of Pennsylvania in the School of Vet Medicine and Pathobiology. Jenny has researched T-cell development for over 3 decades. She's taught immunology, biology, and writing from high school to postgraduate levels in the United States, and is a recipient of multiple teaching awards. She's co-author of the QB Immunology text, if any of you have used that, and numerous scientific articles and chapters. And Jenny's also a section editor with the African Journal of Lab Medicine. We'll also be listening to Marie Chataway. Marie Ann Chataway has been working for the United Kingdom Health Security Agency, which used to be called Public Health England, for 20 years. She's a pathogen lead for Salmonella Services. She's been involved in supporting laboratory capacity development and training, as well as developing workshops for bacterial enteric pathogens for 10 years. This is the second time we're privileged to have Marie contribute to one of our writing workshops. She's been supporting and mentoring international students to write publications and has experience as an editor for another mentoring journal, the Journal of Infection in Developing Countries. She has established an established publication record with over 50 scientific papers in peer-reviewed journals and multiple publications and book chapters. You'll also have the privilege of listening to Elshama Moko. Elshama is an editor with the African Society for Lab Medicine for its publications, and she's also an editorial assistant with the journal. She studied microbiology at Babcock University in Nigeria and went on to learn to earn a master's in science at the University of Ibadan. Her research focuses on bacterial genetics and gene expression. Elshama loves science writing. She began her editorial career in 2017, and she's a recipient of the 2021 Council of Science Editors Scholarship, which is basically the most prestigious training fellowship for editors that I know of. She's currently enrolled in the Council for Scientific Editors Publication Certification Program. We will, as Bethany mentioned, we have a Slack channel where we're doing one-on-one mentoring. You will find, in addition to those that I've introduced, Bethany, who spoke to you just a few minutes ago, who's our managing editor in the communications department at the African Society for Lab Medicine. She currently manages the journal and Lab Culture, which is the society's magazine. In addition to other communication projects. Her formal training is in theoretical linguistics and German, for which she holds a bachelor's degree from the University of New Hampshire. She also holds certificates in from the American Medical Writers Association for medical editing and from the City Program for Human Subjects Research. She's a member of the Council of Science Editors and the International Society for Managing and Technical Editors, and is a past member of the AMWA. We have some offline mentors in this workshop who will be working with you exclusively in Slack. These include Ekesin Odi, who's an editorial assistant with the African Journal for Laboratory Medicine, as well as Pascal Ondua, who's a section editor with the journal and also scientific officer for African Society for Lab Medicine. And then I'd finally like to introduce our first speaker, Dr. Ike Anya. So Ike is a consultant in public health medicine and co-founder of Nigeria Health Watch. It's a great blog. You should check it out, nigeriahealthwatch.com. And Epi Afrik. He has over 15 years experience in clinical medicine and public health in Nigeria and in the UK. He's a fellow of the Faculty of Public Health. He has taught at the London School of Hygiene and Tropical Medicine, Imperial College, University College London, and University of West England and Bristol University. He's co-founder of the Abuja Literary Society, the Public Health Foundation in Nigeria, and is a TED Global Fellow. Ike will be kicking us off by basically asking the question, why publish? And what is the story that you would like to tell? Ike.
Okay. So good evening, everyone, and apologies for that slight technical hitch. As Iruka has said, my name is Ike, and I'm going to be talking about why publish and what's the story you would like to tell. So I assume that everyone who signed up to this session has signed up because they want to publish more research papers. And the question, I think, as a starting point that is always useful to ask is, not just for publication, but for pretty much everything you want to do, is asking yourself, why am I really doing this? And sometimes the answers you give when you ask yourself that question may not tally with the answers you might give if people asked you publicly why you want to do that research. And that's fine, because we all have different kinds of motivation. I'm not suggesting at any point that there are some good reasons or some reasons that are not so good for doing research. Some people will say, I want to publish research so that I can become more noted in my career. I want to publish so that I can progress. For instance, if you're a young lecturer or you're a young PhD student or a newly qualified postdoctoral fellow, if you're in academia, you might want to publish so as to progress your career. Other people might say, well, I want to publish because I think it's a nice thing to be able to say, I had a paper published in so-and-so journal. Other people might be motivated by a genuine desire to share what they have found in their research. And I think a mistake people often make when you talk about why we're doing research is that people assume that you can only publish in
Hello, everybody. We seem to be having some technical difficulties with Ikeanya. So that we don't lose too much time, I'm going to suggest that we actually go over to the second presentation, which is mine. And then if we can bring Ike back after that to finish up, then we can continue with him. Is it possible to have my slides, please? Thank you so much. Thanks. So I'm going to be talking about basic publication etiquette and ethics. Next, please. Next slide, please. So So Ike went into some of the motivations and imperatives to publish. And for some of us who are health workers, it's actually a moral imperative to share things that you have learned because they can impact the health of other people. Next slide, please. However, there's also the issue that we don't want so much information that we can't find the information that's useful. And actually, this is something that came up during the current pandemic. It turns out that the literature on COVID is so humongous that the people who are actually working on COVID cannot actually find the information that they need. So there needs to be a balance here. Next, please. Next slide. Okay. Then, so one of the reasons why there's this deluge of publications, obviously, is because in academia, career development is tied to publication. This is something that Ike was alluding to. It's important to think about what the quality and what the message of what you're publishing actually is. Having very many numerous publications is actually not beneficial, even within academia where we have a publish and perish culture. And then obviously to really write well, you have to read well. And there's a balance between the reading and writing that would determine how much you're able to publish. Bad writing is easy to turn out, but good writing is actually an essential skill for anyone in academia or anyone who needs to communicate. Okay, so it's very important for us to think about whether what we intend to publish is worth it, and this is a large part of what Ike's talk is supposed to is about. But also we have to think about our responsibilities to those who are going to read those publications. Next slide, please. So it turns out that as an author, you have a responsibility to the audience that will read your work to ensure that what you publish is relevant, sincere, and worthwhile. Next slide, please. So the reader has the right to expect that when you publish something in the peer-reviewed literature, that what you're publishing is original, unless you say, oh, well, this is a review or this is a recap. But if you don't say that, if it's a primary research paper, the reader should expect that you are actually publishing something that is new. The reader also has the right to expect that what you're publishing is, as far as you can determine, accurate. So it has to come from the result of work that has been done carefully and reproducibly. And the reader also has the right to expect that when you're presenting ideas of your own, they are actually your ideas. Next slide, please. So all of this requires us to do our science with integrity. And this involves everything from conceiving the idea, building your collaborations, to the actual writing. So there's a number of things that we have to think of when we're designing the experiments that will appear in the scientific literature as papers. I'm not going to go through the whole list, but there's quite a lot. Okay? And when you have done all this, it's also important to write with integrity. Next slide, please. So when I'm talking about integrity in scientific publishing, what am I talking about? First of all, integrity from the author or authors. Okay, at the African Journal for Lab Medicine, our commonest reason for having to reject articles, and this surprised me when I became editor, is actually plagiarism. We don't actually see a lot of plagiarism that is stealing of ideas, which is obviously the worst kind of plagiarism. What we often see is text plagiarism, which is lifting text without properly attributing the source. And I'll go into this in a bit. If you're writing with integrity, your literature review should be unbiased. That is to say, you should not be preferentially referencing sources of people you like or work that you like to the detriment of people you don't like or people who are publishing things that don't agree with your findings. It helps to do a systematic literature review to avoid those types of bias. Very, very, very important to avoid data manipulation or exclusion of various types of data. This is a huge issue in science and one that many journals are now pursuing. Many journals are actually now getting tools that will look at the figures that people include in publications and be able to see whether or not they have been manipulated. And then author contributions. It's very important to make sure that everybody who deserves to be an author is an author, and everybody who does not deserve to be an author is not listed as an author. And I'll go into this in a bit more detail as well. If you have conflicts of interest, say you're writing about a diagnostic tool made by a company that you own or you have shares in, this is something that absolutely has to be declared up front so the reader can have that at the back of their mind as they're reading what you write. And then even after you have published, your responsibilities for what you have published continue. So if you find that there was a mistake, or if you find that one of your co-authors has been dishonest, you still have an obligation to correct that mistake or retract the paper as is necessary. Publishers also have a number of integrity issues to worry about. I won't go into too much detail about them here because that's not the audience for this workshop, but enough for you to know that those things exist. There are journals that have really good intentions, and there are journals that don't have good intentions, so-called predatory journals, whose prey, you know, when you talk of a predator and a prey, it's actually authors. They are the ones that they eat up and destroy. Next slide, please. So I wanted to spend a few minutes talking about plagiarism because I realized that when we point out plagiarism to authors, many of them are actually unaware that they have committed this offense or that plagiarism is indeed an offense. So as I mentioned earlier, if you take somebody else's paper, delete their name and put your name on there, that's obviously plagiarism. Most people understand that. If you take somebody else's ideas and pretend they're yours, that's plagiarism. Most people in science tend to know that. However, if when you're writing your introduction, you assemble a number of sources and pick out blocks of text from these sources and paste them into your introduction, you have actually committed the scientific fraud known as plagiarism. And these days, it's actually very easy for you to be detected if you do this. So it's very, very important to make sure that you're not doing this. It is scientific fraud. If you're detected as somebody who plagiarizes, you can be blacklisted. Your reputation can be tarnished even before you know that you've done something wrong. And, you know, I tell my students, because this is not just a problem that we see in scientific publishing, we also see it in academia. I tell students who inadvertently plagiarize that one of the things they're losing is the ability to learn how to write. Because if you write badly, you can be given feedback that makes you write better. But if you paste in somebody else's text, then nobody knows that you need that feedback. Okay? So I'd like to encourage people, please, do your own writing. If you're using sources to get material for your introduction, the rule of thumb is you read the source, you close the source, and then you write. Next slide, please. So, who is an author? It turns out that not everybody who contributed to the development of a manuscript is actually an author. And I'd like you to think about some of the categories that I've placed on this slide. And as you're thinking about them, I'll also tell our applicants that applied and were accepted to the interactive portion of this workshop. If you're not already on Slack, please join. So we will paste these questions on Slack and have you play with them. If, like me, you're having trouble keeping up with Slack and keeping up with the live session, don't worry about it. The Slack will be open until Thursday. It also means that your writing mentors may be a little slow to respond to you on Slack while they're talking, but we will get to you before Thursday. So think about these different categories and including other candidates, your favorite author candidate, and think about whether or not they are actually authors. And to help you with this, next slide, please. We actually, those of us in the Council for Scientific Editors, we have a guide that we use that helps us determine whether somebody merits authorship. So if you've done more than one of the things on this list, there's a good chance that you're actually an author of that paper. Okay? If you've done one, then it's a big maybe. If you've done none, you're not an author. If you've done 3 or 4, you're probably an author. And it's important to mention that it's not only a question of deciding, who worked with me that should be included, but also for you to know when you're contributing to somebody else's project, whether or not you deserve authorship, and whether you should be having a discussion with this collaborator about that. These discussions are best had in advance before a manuscript is compiled. Once a manuscript is submitted to a journal, it becomes very difficult, not impossible, but difficult to change the author list. Next, please. So if you are, I'll just say that after this, I only have a couple more slides. So just to say that if you're planning to write a manuscript that's going to be submitted to peer review, some of the things to have at the back of your mind is that you need to learn how to write. Now, unfortunately, certainly in Nigeria, our high schools tell us, well, if you can write, go into the humanities and law. If you're not too good at writing, but you're good with numbers, go into science. This is rubbish. Okay? If you are going to be a scientist, you still need to learn how to write. So learn to write as early as possible in your career. It's very, very important. If you're doing a literature review, make it systematic, even if that's not required. The Prisma guidelines for systematic reviews, which we will post later in the Slack channel, are helpful guidelines for figuring out how to make your literature review less biased. Okay? Read your primary sources. I find so many authors just read the abstracts. Very important if you're citing any reference to have read the whole paper from beginning to end. Cite it correctly. Data included in your manuscript. All authors who are co-authoring that paper are responsible for its content and have to take responsibility for the validity of the data. If you think you can't do that, then you should not be an author. Okay, and then ensure that everyone who should be an author is an author and everybody who should be acknowledged is acknowledged. And write your work up ethically and well. Okay, next please. So I'm going to stop here. We're not going to take any questions right now because after all the presentations, we will have a panel discussion with all the presenters. I'm going to hand over to Jenny, who is going to be talking about background, hypothesis, and aims. And after that, there'll be a series of talks on different parts of the manuscript. I won't come back to introduce each one. Jenny, thank you.
Thank you. And I think everybody can hear me. It's really a pleasure to be here. An honor to be here. I'm from Philadelphia, and I'm looking out at a beautiful blue sky here. I know some of you it's evening, and some of you it's maybe earlier afternoon, and welcome to all. I have been writing with students for a long time. I've written my own papers, of course, and I review papers. And I just want to reiterate something that I think it was Aruka, it might have been Ike who said it, that the best writers are readers. And reading the entire paper is very important, but as you read, you will learn better and better how to write. And writing is a lifelong effort. None of us are perfect at it, and I know that over the decades, I've gotten better at it. And you can think you're a good writer when you're in college, you're going to get better. And as you get better in the scientific world, you'll see that you change with time. So I'm going to focus on your introduction, which really comes out of the research that you've done, your background, your significance, your question, hypothesis, and objectives. And one of the first things I thought that I wanted to convey to you and I convey to my students is that good papers actually have a shape. They have a shape where you want to position your audience, center them in the field of interest. So you're bringing them in and then focusing them on your particular question and your hypothesis. And other people here are going to talk to you about how to do the rest. The other part of the shape after the focus is that your discussion should bring your audience back into the field of interest and help them to see what gaps in knowledge that you have fulfilled. It is, as EK said, it's a narrative. It's a story that you want to tell. The what and the why is very important, and you want to identify what is known and what isn't known so that when they get to your question hypothesis, it's relevant to them. Okay, so how do you write this strong introduction? Many people will have really good tips. I will share some of them. They're not going to be the only ones, and some of them will resonate more to you. I have put some things in the chat and Slack. This website is quite useful too, and it's something that's even useful for an experienced writer. The first thing that's really relevant to what EK said, know your audience. And to what Aruka said, I think you've got to know who your readers are. If they're really focused experts in your field, there are certain things you don't need to include in your introduction. But if they're more general, depending on your journal and what you're talking about, you want to center them on the ideas that are going to be most relevant for what you're going to talk about. Your opening paragraph typically describes the broader context. Context is important. You want to bring your audience into the issue that you're going to be addressing. And you need to describe why, the motivation for your study, not necessarily personal, although that's always very interesting to readers, but in a professional setting, the readers need to know what isn't known in the field and what your study will contribute to. What is the motivation for it? What is the original foundations for this? And what are you going to advance? You need to clearly state your question, which I'll say in the next slide is probably one of the most important pieces of your introduction. Your objectives, what you're going to do to address that question, and ideally a hypothesis. And we're going to talk a little bit more about hypothesis generation and conveyance. Ideally for your paragraphs, and this is something that's really hard for some students, and it's even hard for those of us who are professors, cover 1.1 argument per paragraph, so that if you were looking at your introduction, you could actually see it outlined. And this 1 point per paragraph really helps because as you develop this, you should be paying attention to flow. You want to walk your readers down what we call a garden path to your point. And you might find that one paragraph belongs earlier or another paragraph belongs later. And if you keep 1 point per argument, it's easy to shuffle and more easy to develop the flow that you will need. And stay focused. We're all experts, and you as students even more so, or you as masters. I see some of you are masters, some of you are advanced, and some of you are just beginning. You know more about certain things in the world than anybody else. So it's tempting to talk about everything you know. It's very important. Think of your audience, go back, know your audience, think of them as readers, and stay focused. That helps a great deal. So what about this question hypothesis? The question that you're framing is probably the most important. You want to be asking a question that's original, as Aruka has said, and a question that is clearly conveyed so that people can leave your paper, leave your manuscript knowing what you asked and how you answered it. They might decide that you haven't answered it perfectly, but they should know exactly what you asked, and they might decide that you've answered it beautifully. Your hypotheses are very powerful, and they should have two features. They should be testable with the methods that you're going to describe later in your paper, and they should predict something. They should have a certain amount of power, predictive power, and usually predictive about a relationship between two or more variables. And something I always have to remind my students, they don't have to be correct to have an impact. Hypotheses are meant to be testable, to be predictive, to move a field forward. But if they're not correct, sometimes the paper even has more impact and you have a better story to tell. You don't want to aim to not be correct, but you shouldn't be afraid of hypotheses not being correct. Your objectives and your aims should be very, very clear. How are you going to address this hypothesis? How are you going to address this question? And that should be in your introduction. Describe what you're going to do this. And there are some words that I find are very helpful in a paper, whether you include them explicitly or whether they're implicit. It's always good to remind yourselves as you move through your results, but also even in your introduction. In order to do something, we embarked upon this. to address this question or to address this problem, we did this. And those things really help with flow, and they help also center you and focus you on what you really want to convey. An important thing that's come up, especially these days when we have these very high dimensional data, that's really important, is that not all research is hypothesis driven, and it doesn't have to be. Some is discovery based, some, as in the African Journal of Laboratory Medicine, can be based on desires to improve safety, to do things. However, I will even argue that even discovery based research and even things that are describing new techniques have some underlying hypothesis that could be powerful. And at the very least, they have objectives that should be clear. I always try to have my students, even if we do discovery things, to really think about thoughtful predictions that do help drive and clarify some of the things you want to do next. So Verb Choice, I'm not going to go over all of these, but I really do like this website in particular. And the Verb Choice helps. We used to write in more passive voice, and I suspect some of you are still being taught to write in passive voice in certain contexts. But most of the time, we're shifting to the active voice and using verbs that really help, are really illustrative, really help. And they help propel your reader through your intro as well as other parts of your paper. And there are just some here. We determined, we developed, we investigated, we measured, we modeled. These things help. And they help even me, as experienced or so-called experienced as we are, in this lifelong quest to be a better writer. Developing the hypothesis. That can be both the most fun and most challenging part. I borrowed this slide from a colleague who I will cite at the very end appropriately, and with ethics. But he has a very wonderful way of talking about how to develop hypotheses. And we work with both master's students and PhD students along this. It's really an iterative process. It's not a single an easy route to develop it. You're asking your question. You might even change your question as you do your preliminary research. And then you start saying, okay, here's my prediction. It's an educated guess that is testable and can move a field forward, but you need to refine it. And then when you start to convey it on your paper, you actually should work on phrasing it in different ways. Changing the phrasing can actually help you go back and do some other research. So do not be afraid to, don't get married to your hypothesis. Do not be afraid to change how you think about it and how you write it. That kind of iterative process is part of writing well. Some people say that to develop a hypothesis, you should do like an if-then statement. And I had some examples, like if we introduce a new diagnostic test, then we will reduce incidence of a disease. That's one way to do it. But there are other ways to generate hypotheses. I have another example, the ability to generate antigen-specific antibodies diminish with age. That's not necessarily an if-then statement, but it has two variables in it, and you're testing one. So that's one hint. There are many others that I'm sure your experts will help you with. And next. So I can talk at you, but I wanted to give you two examples of recent papers where I think they both have effective introductory sections. But one of them, the next one, I think still has a little bit of a hole in it. This one, which I think is quite good, is from the Annals of Internal Medicine about resistance testing for management of HIV virologic failure. And they start beautifully. They provide context. Virologic failure in HIV remains a major public health threat. They give you a little bit of background about it, what percentage of patients and how long after initiation it happens. And then they identify the problem. This is already a problem that's implicit, but identify it very clearly. And the unknown, the optimal management of virologic failure is unknown. They have actually told the reader what is unknown. They describe a test or an approach called genotypic resistance testing that might be useful, that might help reduce these failures. And then just before they go into their results, they have a beautiful paragraph where their objectives, their hypothesis, and approach are all there in 3 sentences. We did this, an unblinded randomized controlled trial. The trial aimed to, their objectives are to estimate the effectiveness, and they hypothesize the addition of this to routine care would improve rates. So even though it's not exactly in the order that I discussed, it's still very clear, and you don't have to have perfect order for this. I thought this was a lovely example, and it was a very short introduction, succinct and useful. The next one I think is also very clear, but they're missing something. So this is also in October 2021 in the New England Journal of Medicine. The audience are going to be doctors as well as scientists, young and old. And here they present their problem: vaccination of individual persons for for SARS-CoV-2. Whether vaccination protects members of their households is unclear. They identified the unknown. They talk about the broader context, and then they talk about what they did. We investigated the effect of vaccinations of healthcare workers in Scotland on their households, on transmission rate in the households. But what's missing there, and you can say it's not that important, but it is a way to identify what might be missing, is their hypothesis, their objectives. They're not saying it clearly. They could easily have said, we are looking to see whether vaccination of individual persons will protect members of their households. That is their objective. Instead, they ask you as a reader to interpret that. It's not a big leap, so I don't think it's really a huge problem, but it is an issue. And in their last sentence, their last paragraph, I think it's also clearly written, and you can take a look at it. But they dance around their hypothesis, which you as a reader could easily have written for them. Again, I think they're still very clear, so I'm not going to be too critical about it. And you can take a look at it when you see the PowerPoints. So this is just the last bit, and I very look forward to answering questions and listening to my peers, because I'm learning from them all the time. Acknowledgements are important. Citations are important, just as Aruka said. Tom Parsons is my colleague. We work together with master's students on a research master's, and he has a beautiful way of describing multiple kinds of hypotheses, how to generate them, and how to convey them. So I give him a lot of credit. These two websites I found very useful. For tips for writing a scientific paper. There's several other ones on Slack that are very useful. And I think perhaps the most useful feedback that you can get is from your peers. I know how hard it is to share your work with others. Critique is hard. It's really hard to receive. It's hard to give too, but I wanted to thank all my teachers, my professors, my mentors, my colleagues who've had the patience to provide the feedback, to read those drafts. Do it, be generous to others and read drafts. You'll learn from it, and they really can give you some of the best feedback. And peers are perhaps the most important. They will be honest and say, this doesn't make sense. I don't understand this. And take it with a generous heart too, because it'll really help you read your papers. So that's all I have for you. I look forward to questions, and I will hand it over to our next speaker.
Hi everybody, hopefully you can hear me. I'm Marie Chataway. I'll be talking about results and figures. This is quite an interactive presentation, so I'm just going to pop on Slack to see who has access. If not, I'll be asking the questions, and I would like people to be making notes, because historically when we do this on site, people will shout the answers out to me. So we'll have a look and see how we get on. So if I go to Slack, there's a section called the Manuscript Writing Workshop. If people can just quickly post a hello in there, just so I can see how many people have access from the group that we've set up with this, that would be really useful. And if nobody posts on there, then I know that nobody's looking at the moment. Okay, great. We have one person. Anyone else? Okay, that's fine. So Aruka did mention that it is quite hard to do live and do Slack at the same time. So what I would suggest then, when I ask these questions, I'm going to pause for a few seconds, and I just want you to have a think and just jot some things down. Okay, so if we get a start with this, the results. Okay, so I do apologise. The presentation that I put up usually it comes in parts, but I can see from the way it's set up on this that everything comes up at once. So you've got the answers already there. So what I tend to ask people is about the common errors that scientists present when they're doing results, and the common sort of results issues that people get is sometimes they will go back and they'll start redescribing their methods again when it's not results. They may be starting to have opinions, interpretation of the results, rather than specifying what the results are, which should come a bit later in your discussion. Sometimes they'll repeat results, they'll write things in text and then they'll write them again in figures. They may go back, quite common things where they'll start reintroducing background, and it's like a bit more of a introduction again rather than specifying on results. Also, we see examples where people will start describing results that's not even in the methods, and that's quite a common thing that I see as well. Sometimes just too much text, not being clear and succinct, and also perhaps not reporting all of the data in your results section that you spoke about in the methods. So a lot of what I'm talking about does tie in with what's been described by the previous presenters. And again, it's all about being clear and being specific in each section. And as Jenny and Aruk and other people have mentioned, we have posted quite a few things in Slack to give you some guidance, but the different sections when you're writing a manuscript, it's very important to stay within those sections. You've got your introduction, which has just been described, and now we're talking about results. So the do's and don'ts of presenting your results: do be quite clear, ensure that all your results are presented as I mentioned previously. Don't waffle, and like Aruka mentioned in her presentation, the quality is better than quantity, so that will overarch rather than just sort of waffling on. Have good figures with your results as well. A picture can say 1,000 words, they say, and it's very true. A very good figure can, and we'll go on to that in a couple slides, give you a lot of information about your results, so you don't have pages and pages of description. And have very clear headings as well, and have things structured when you're doing your results as to what you want to present. The don'ts: don't repeat results in a full sort of paragraph and then have exactly the same information in a table or a figure. The idea is that your figures and tables will be complementing your results, and perhaps the results might be a summary, but you do need to make sure that all the information is there. Don't interpretate the data in too much depth or commentary. That will come on for your discussion a bit later on, though you can give some key results or key findings within the results section. And don't present data that doesn't necessarily link into your methods or your introduction. As just Jenny mentioned, it's very clear that you're telling a story in your introduction. Like, what is this study about? Why are we doing it? And your results and your methods should be following on from that. So we're going to go and give a couple examples of how we can visually present the data. So here's an example of how to present the results in terms of demographics. And again, I do apologise that all the answers have come up. Next time I will make sure that they're on separate slides, but unfortunately it hasn't kept it separate. So one of the questions I had was, when you've got demographics such as sex and age, how can you present this type of data? And quite often people might do bar graphs, or they might summarize in a text, there was this many people of this sex and this age. What you can see up here is called a pyramid. It's a sex age pyramid, and here you have a presentation of males on one side, females on the other, as well as the number of cases at the bottom and also the age ranges at the top. So it gives you quite a lot of information looking at that, and you can look at the demographics, and you can see in this particular example that it's mainly the adults and also sort of teenagers that have the highest number of cases. But in terms of male and female, the demographics is quite similar. It isn't anything that's obviously different. Another example presenting data is thinking about illustrative maps, particularly when you're looking at a global picture, for example. And a good way to that is to have sort of map, as we can see here. And you can get something called like a world map, and you can just basically color code it using publicly available tools like Inkscape, and it's a nice way to show and summarize your data. For this particular graph, I used like a world map, and also I used Excel. So in Excel, I did these little sort of pie charts you can see here, and then copy and pasted them onto the world map. and also color coded them by the pathogen. So you have a look, you've got a global world map here. You can see the different type of pathogen and the number of cases and also what countries they're from. So this is a very good way of showing a lot of complex information in a particular figure. And that's something that you need to think about when you're presenting your results. What the type of information I look at and how can I illustrate them? So the other type of results are graphs. Quite often people will do graphs. Pie chart these days tend to be a bit of a no-no. They're quite basic. They don't give you too much information, but presenting graphs visually, again, can give you lots of types of information. So at the top here, we can see a variety of different virulent subtypes of E. coli non-Y57 that was looked at in 2014. On the bottom left, you can see that there is a demographic sort of chart in terms of cases, those that basically had attended a restaurant and those that didn't attend a restaurant, and therefore how many of the cases actually attended this restaurant. And this is something that you might want to look at if you're looking at epidemiology, for example. And on the right hand side, we have a bar graph looking at resistant trends of different antimicrobial resistance. Now, having a look at these figures, what do you think is sort of missing from these figures? I'll ask the question again in the next one. So if these were presented in a manuscript, what do you think would be missing? Okay, so this is another example of a table, but it's also sort of like a heat map as well. And this particular one is looking at virulence genes against certain type of cases. So on the right hand side, you've got the information, what sequence type, whether it's from the UK, for example, was it found from urine and blood, and also any antimicrobial resistance. And this was a particular figure looking to map to see if we could see differences in virulence factors from E. coli that were isolated from blood and stools, for example. Now, I've got the answer on the bottom, unfortunately, but basically what is missing from all these figures is a descriptive footnote. And this is something that's going to come up in exercise 2, which we're going to post in Slack, where we will post a figure and we want you to write a descriptive footnote. And it's quite important writing a good descriptive footnote, because the idea of having a good figure in your manuscript is that when the person is reading the manuscript, they should be able to understand what this data is presenting and summarizing without having to go and read all the text. So in a way, it should be independent of what you've got in the text. It should complement it, but people and the readers should be able to read it, read a good footnote and understand what this figure is showing without having to try and work it out from the manuscript text. If you can't do that, it means it's not a very good descriptive footnote. And sometimes as well, people put sort of a brief summary of the main data set. So we have an example here. You can see the original footnote just says trends of resistance. And so that's a very poor footnote. It doesn't give you any information on what the organism is, what the geography is, the year isn't stated, we don't understand the acronyms. It's just not very clear at all. And then underneath you can see an example of a better footnote, and I've tried to color code the additions that's been added to the poor footnote. So for example, you can now see we're talking about Salmonella typhi, that we looked at it within 2016 to 19, that actually it's been categorized and it's divided into main categories, and that you can see the trends. And at the bottom you can see there's just one little result line summarizing the key point of this figure, saying that actually it shows an increase of resistant typhi, with the largest increase in 2019 and associated travel to Pakistan. So that gives you a lot of information. And that's ideally what you want when you're thinking about describing your results and figures. So I'll just go through a couple more figures because I've just got 5 minutes left. So a couple examples here. This is epidemiology. This is a sort of an early figure, and this is when they were tracing Vibrio cholerae. And what he did back then was map all the deaths in a particular postcode, and that's how they worked out that there was a particular pump associated with all these cases. On the right, you can see an example of a phylogenetic tree, and it's been isolated from food and restaurant and cases. So it gives you an understanding of looking at that. So results and repositories, what's quite important with your results is you need to make them publicly available, whether it's sequence data or protein or anything like that. And within these presentations, and I understand these are going to be shared, there are some examples of the different type of repositories that you can put your results in. When you've got very complex and long results, you quite often have something called a supplementary table, which will have all your raw data. And obviously the figures you have in your main manuscript will be summarizing your results. So for the discussion, again, similar to the results, there are common errors that scientists tend to do. I won't go through each one individually, but the most common things that I see are people start redescribing the results all again. It's okay to mention them as part of your interpretation and discussion, but you shouldn't be repeating all of that information. Poor flow, will people jump about? And this again comes to what Jenny was saying. It's quite important that you have a good flow. It's a story in your manuscript, and in the way the flow should be matching from your introduction to your methods, your results, and then on to your discussion. Quite often people will start repeating a whole background introduction again. You should be referring your results to literature and integrating your interpretation, but you shouldn't be doing another introduction of just talking about what had been seen before without tying it into what your study is about. And again, not too much text, not too much waffling. So what should we be doing in a discussion? So you should be summarizing what was found. Quite often it's good to put your key point or message in terms of your discussion, especially when you've got multiple different types of results, which you will have for a lot of your complex studies. Make points. Again, it comes out to structuring your points. Talk about your strengths and limitations. Aruka spoke about not being too biased in terms of literature review. It's the same as well of interpretation. You'll get some studies that will complement your study, the others that won't. And it's okay to say there's conflicting results. And also at the end, have very specific conclusions, recommendations of the impact of your study. So for writing your discussion, I find it quite useful first to start off with a structure. Do bullet points, the key things you want to get across in your discussion. It's quite useful sometimes to start off with all the different subheadings. You can always take these out in the end, because when you do read manuscripts, you can see that you don't often get subheadings. Sometimes you do, but it's useful to start off with that. Again, quality over quantity. Don't waffle, be clear and concise, and you should have that flow throughout your entire manuscript. Now, in your discussion, it's important you're not introducing new information, whether it's results or you're talking about something that you haven't even mentioned before in an introduction. So it's quite important that you keep it relevant to what your hypothesis is, which Jenny mentioned at the beginning. And ideally, look at your objectives and talk about those and make sure that it does relate. So again, you speak clearly and consistent. And at the end, the reader should have quite an understanding of your study. They should understand what's novel. They should understand why it's important, which again is something to reiterate in your discussion. And also, what is the impact of your study? Because if there's going to be no impact at all, then actually, is it publishable? And I've got some information here talking about what I've just mentioned, and if your study doesn't cover any of those concepts, then it's unlikely that your actual study itself is worth publicizing. So I've put any questions, but of course we're going to interact later on in the panel. We can also post any additional questions that we have in Slack, and like Aruka said, it's going to be online for a couple of days, so we can always sort of come back to that. So that's the end of my talk, and I'm going to hand back to Aruka, but I think we have a little bit of a break now. Aruka?
Hello, everybody. We're going to skip the break to catch up on time, and we'll have Ashama and Moko giving us the next talk, which will be your own copy editor. avoiding the presentation errors that can get in the way of peer review. Shama.
Hello everyone and welcome to this session. So I'll be taking you on some errors that you must avoid to make your manuscript publication ready. Next slide please. Copy editing essentially tries to make your sentence, the intended meaning of your sentence clear. It also helps to ensure that your formatting is consistent as well as your language, and that your data and your facts are correct and the parts of your manuscript are complete. So you can see copy editing more or less as fault finding. Next slide, please. So, yeah, next. So we'll be looking at the errors that particularly interfere with the completeness or the inconsistencies in your manuscript. Next slide. So generally, every journal has their instruction for authors. You have a preferred referencing style, a preferred abstract format, manuscript format, guidelines for illustrations, and word limit. Try as much as possible to know those instructions. For me, as an editor, I'll be looking at, have you exceeded your word count? Is your abstract structured according to our instructions? So you need to know what journal wants from you and then implement these. Then another general error I commonly encounter is authors not providing citations for major claims methods in their manuscript. Next slide. So previous authors have mentioned some of the components of the manuscript. You have the title, the abstract, and all the others that make up the framework. So I'll be going first. Next slide. I'll be going first with the common error with the title. The common error with titles is that they are uninformative. And when the scientist reads your manuscript because they have a name, so your title should be able to inform the reader that if your research might meet their reading needs. So it should have the certain location, your participant ID, from the intervention, the comparative, the main outcome, the period of duration, and sometimes the design of your study, like the example we have in the slide. Next slide. Next slide, please. Moving on to the abstract. I commonly see people not including their main method, or missing out on the study period, the duration, the sample size. These are salient points which the reader needs to see to confirm that your manuscript contains what he thought your title has referred. Also, most people just repeat their results in their conclusion instead of highlighting the implications of the findings. And then the most upsetting of them that I encounter is when your result, the abstract, is different from what you have in your conclusion. So try as much as possible to write your main text first before writing your abstract. That way, there won't be inconsistency in your data and your results. Also, keyword is one of the things most authors don't take too much interest in. You just randomly put whatever they feel like putting there, but you need to, the keyword tells you the scope, tells the reader the scope of your research, and so you need to think carefully about the scope of your work to do right keywords. Next slide. Next slide, please. Yes, for the introduction, Jenny has mentioned a lot of things that you need to put there. And I'd just like to reiterate that oftentimes, most introductions just have a weak theme, and the problem might be described. However, the justification of the work as well as the aim is left to the reader's assumption. And that is one thing that you need to avoid when you're writing your introduction. So for me, as a copy editor, most times it's almost like a checklist that you're going through. So when you're going through the introduction and you don't see some of these statements in clear terms, it can get in the way of your next slide. Next slide. This is just an example of an introduction that has some of the components that we'll be looking at: context, justification, aim, approach, and sometimes it tells the reader what to expect going into manuscript. So we enrolled 1,000 women in our research, and we found just a brief intro to your results. Next slide, please. Then in the method, there are a lot of errors that we encounter as well. Most authors don't clearly state the ethics of their research. They feel sometimes that it's enough to just put the ethical approval number and the review board, but you need to include how you got consent from your participants, how the privacy of their data was kept, as well as how you identify your data. So when we are going through your manuscript, we'll be looking at all these components there. If not, then it raises issues with credibility. Also, sometimes this research design is often poorly described, missing so much information that is necessary to explain what took place in the research. Things like the study type, the nature of the participants, the testing method, and particularly how your variables were defined. If your study is a study on diabetes, how did you define diabetes? I could define it as just having sugar in my body. I don't know what anybody could have any definition for diabetes. So you need to state the definition for all your variables, and also how did you minimize errors for your for your measurements. Did you do your measurements in duplicates? The sample size, how did you calculate it? Did you blind your participants to minimize bias? How did you minimize error in your manuscript? Those are part of the things you need to look at while you're writing your methodology. Don't leave anything to intention. Also, your statistical analysis. Most times people just realize, oh, we did we use test, we use features exact, we did linear. Whatever it is you're doing, you need to provide a statement that tells us that your data met the assumptions of the test that you are using. And then you can also state the significance level and confidence interval. Next slide, please. Next slide. Right. Then for your results, I'll start with the table because that's the first thing you compute when you're gathering your data while you're doing your experiment. So when you want to present a table for publication, the first question you need to ask yourself is this table necessary? Is it more efficient than words? And is it just a storage of data? It must be able to tell something better than words, and it shouldn't overburden the reader. So when I see a table that has just two columns and two rows, you can put that in words. Then you also need to organize your rows and put your variables in the same order. Tell us the units of your of measurement. Also, one recurrent error is when your data is not reliable. When we total the columns and the rows, are they accurate? A simple way is to use Excel or any other automated any other software that can automate your calculations for you. And then you'll be asking, does it have enough information for me to understand this table. As a copy editor, I'm like, I'm neither a reader, I'm neither an author. So I need to be able to understand your table without going back to your manuscript to understand what you're putting. And then sometimes the title of the table is always uninformative. An informative title tells you the topic of the variable that your table wants to show. It also tells when the study was done. It also tells in whom it was done. These three things are very important. And if you're comparing variables, you also need to put those variables. For example, if you're saying zinc reduced the episode of diarrhea, so that needs to be on your title so that when I'm reading your manuscript, I'll be able to know, oh, this table is not for me to read, this table is not for me to read. So those are the common errors I see in tables. Next slide. And then everybody, almost all the speakers have mentioned this. It must be comprehensible without referring to the main text. Next slide. Then moving on to the figures. Sometimes you have to provide figures, but the question you need to ask yourself is this figure necessary? Can I explain the process without the figure? If yes, no need for the figure. If no, then put the figure. And if there's a need for the figure, it must be organized and reliable. This applies to flowcharts. Sometimes you need to tell how participants were excluded from your study. So your numbers must tally on your chart, and that makes it more reliable. You also need to provide a footnote describing whatever it is that is on your figure and an informative caption. Next slide. And it must be comprehensible without referring to the main text. Next slide. Next slide, please. For the graphs, yes, sometimes you need to show some trends. The question is, can your reader notice the trend without the graph? If yes, no need. If no, then put the graph. And then, is your graph appropriate? Like Mary said, some people, sometimes you could use the pie chart, whereas you should have used the bar chart, or you use a bar chart, whereas you should have used the line chart. Is your design interfering with readability? Sometimes the background is too dark, the data that inspires are not clear enough for reader. So are your axes labeled, and is the unit of measurement there? Is your base 0? If not, you need to explain why, or start with 0 and put a break in your graph. So, and then you should also be able to provide a footnote explaining symbols and abbreviations on your graph, as well as providing a good informative next slide. Next slide. And for the third time, it must be comprehensible without referring to the main text. It's a lot of work reading an article. If I'm writing a review and I'm reviewing like 200 articles, I don't want to be jumping between text and images to understand what you're saying. So your figures must be able to be comprehensible without the main text. Next slide. Next slide, please. When moving on to your text, your result text, sometimes authors try to skim over their primary findings. Your primary finding could be different from the original finding of your result. For instance, I'm collecting samples in a rural setting and it was difficult for me to continue with the research. I need to state that, for example, in my setting and in one of our research, parents did not want to enroll in the study because of some diabolical beliefs. That is a primary finding. It could help any other person that is doing a research in that setting put that into consideration while planning their research. So always put your primary findings, the total number of patients enrolled, and stuff like that. You also need to, another one is replicating data, like Marie said, you don't need to replicate the data. Auto reference is a very common one. Sometimes tables are not even referenced in the main text, or they are referenced non-sequentially as they appear, or sometimes the authors just repeat the title of the figure or image table, and other times just use the figure states which doesn't provide any information. So when you're referring to your text, highlight whatever it is you need the reader to go and get further information on in your table and figures. Next slide. Next slide. Another issue with results that we see is miscalculation attempts to emphasize that you need to be sure that your figures, your data is accurate. Sometimes when authors are referring to, you need to state the units of your measurement. For example, we isolated 15 E. coli, or we isolated 15 E. coli strains from 20. You need to tell us what your units are. Sometimes we also see that authors are silent on some data that is either missing or they are outliers. And that really downplays your integrity as a researcher. So in either way, it's a lose for you. So you need to put, you need to be sure that your result is accurate and you are not missing anything, right? Then going on to the statistics, sometimes, like I mentioned earlier, we use the wrong statistics, and the one that is often misused and abused is the mean, because the authors don't know when to use a particular statistic. Take, for instance, when your data is not normally distributed, you need to preferably use a median than a mean to describe that, because the median is more resistant to skewness, and also you need to provide the ranges of your data. You don't just say, oh, the average age for participants were 12 years. What is the beginning? What is the end? If you're not giving the actual ranges, you should also provide the interquartile ranges. And when you're presenting your statistical p-value, you also need to be sure that it is relevant at that point. Sometimes for a random study, presenting a p-value might imply that something is actually statistically significant, whereas it was just chance. So sometimes p-values might not be useful, so you need to know when to apply them and when not. Next slide. Still on statistics. Next slide, please. Still on statistics is the p-value. So most people just put the p-value without telling us what was the actual difference between the two groups. What is the range of the difference? What is your confidence interval? Because this is very important for clinicians to know if the finding was actually significant. The difference in two episodes of, let's say, a heart attack might not be relevant. It might not be clinically significant compared to the difference in, let's say, 20 episodes of heart attack. And then you need to actually state your actual p-value. This, for example, if you have 0.49 and you have 0.51, depending on what my assumptions were before I started my research, I could say, oh, it's actually significant. I don't care whether it was within, it's close to 0.5 or not. Next slide. I'll stop with the discussion. Your discussion, please go back. Your discussion needs to have some things that are salient. It must be able to remind us of your aim, and it should be able to compare your results. But one error I commonly see is that people just roll out studies without telling us what is the implication of their result findings with those studies. So you need to not just list studies that are similar to yours, but tell us how they relate to those populations and the implications of your findings. And lastly, you have to critique your work. It's not nice reading, nobody is perfect. And of course, our work might not be perfect, will not be perfect. So you need to state the strengths and limitations of your study. If you don't state that, it tells, it can be assumed that you don't think that your readers are smart enough, or you think that you're too smart, and it downplays your credibility as an author. And then you can now summarize all of this as your small conclusion at the end of your manuscript. Those are basically some of the errors. In the Slack, we discuss how to now work on your text that is now complete, consistent, on how to improve the sentence structure and clarity of your text. That will be all from me, as my time is up. Thank you very much.
Thanks, Elshama, and thanks to all our speakers for some really nice talks on the many components of manuscript writing. I'm going to give a short and final presentation on seeing your manuscript through peer review. Before I start, I'd be grateful if I could have my slides up, please. But before I start, I did want to ask participants, if you don't already know this, whether or not you're pre-registered for this workshop, you can put questions in the Q&A for the platform. And those questions are coming up in the Q&A. And even if you don't have a question, you can actually look at the questions that are posted and vote up the ones that you would most like answered by the panel. If you are registered on the platform, you can ask additional questions on the platform in Slack. And you'll also see that we've posted a number of exercises for you, which we're hoping that you'll be able to work on over the next couple of days, and we'll provide you with feedback through that platform. So I'm going to spend just a few minutes talking about how you select the journal you're going to submit this fantastic manuscript that you've put together, and what you have to think about while you're seeing that manuscript through peer review. Next slide, please. So first of all, it's really important to find the right journal. So, you know, Jenny was saying know your audience. But the nice thing about submitting an article to a journal is that you have some power to choose your audience. And so if you would like your paper to be addressed, for example, to people who only work in laboratory medicine, then a laboratory medicine journal is appropriate. On the other hand, if you wanted to reach a broader audience, you may have to publish in a journal that has a broader remit. Even within and between and around fields, there are other considerations to pay attention to. If there are no articles that look like yours or discuss the same subject in the journal you're considering, there's a good chance that your article is out of scope. You don't want to be submitting your article to a journal that isn't within scope, because that is a very common reason for rejection. So you want to make sure that the journal is in the field that you are writing in. The other thing is that publishing journals that you read, you'll understand the journal and what your article should look like, and you can guarantee that other people will be reading it too. You want to make sure that people who you want to read your work are actually reading that journal. And then you also want to make sure that you're publishing in a good quality journal. And there are varying qualities of journals. Some of them are actually, as I mentioned before, some of them are actually deliberately trying to hurt authors, the so-called predatory journals. And there's some journals that are just publications that are learning how to publish. If you're an established researcher, you may decide that you want to publish in such a journal in order to boost it. But if you're a new investigator, you really want to publish in a journal that's established, that will be read, and also that has a kind of editorial team that can help strengthen your manuscript through peer review. Next slide, please. So, as I mentioned, it can be very confusing. If you have mentors, ask them, what would be a good place to put my journal? If you're a good reader, you'll have a good idea. You'll see where many of the articles that you're working with are published. And then you can actually choose to publish in those journals. There are society journals. For example, the African Journal for Lab Medicine is the journal of the African Society for Lab Medicine that is hosting this conference. If you're a member of the society, very often you work within that area, and the society journal might be a good place to put your work. If you're concerned that your audience may not be able to afford subscriptions, and this is true for many of us doing science in Africa, then considering an open access journal is a good idea. Some people are afraid of author fees from open access journals, but do be aware that many open access journals that do charge author fees will grant a waiver if you are an investigator working in a resource limited setting. So you can read the author guidelines and small print in the journal and find out whether a waiver could be granted to you. And that might be a way to access such a journal. Just to say that the African Journal for Lab Medicine, in addition to being open access, also does not charge any author fees. But that's actually rare. Most journals are one or the others, or the other. If what you're publishing is interesting but not necessarily novel, you could consider a technical magazine. It's not a peer-reviewed publication. It won't count for the same thing for your academic promotion, but it's still an organ for you to be able to disseminate information in. So a magazine like Lab Culture, for example, that's ASLM's magazine, might be a place to put something that isn't necessarily a new discovery. What you want to avoid with all your power is predatory journals. Predatory journals will typically charge you a large author fee, but will not do the editorial work that ensures that your journal is good quality, archived properly, and presented properly to your readers. So you want to make sure that you're not publishing in a predatory journal. A few red flags: if the journal is asking you for fees before peer review, chances are it's predatory. If the journal says we can get your paper turned around in 4 days, a week, you can be sure it's a predatory journal. Most journals will need at least 3 weeks to get something through peer review, and sometimes much longer. Okay, and again, if you see grammatical errors in papers in the journal, things are not formatted right, that's your cue that this may be a predatory journal or maybe a poor quality journal. Very often people who don't have a lot of confidence in their work because they're new to writing want to go for, what one might describe as a lower impact journal. I advise you, go for the best journal that could possibly accept your work. If it doesn't get accepted, the peer review process is still very, very useful. And there's a tool online known as jane.biosemantics.org, that if you paste in your title and your abstract, it'll actually tell you journals that are in that area, and what their relative reputations are and so on. So it will give you a list of journals that you could consider. Next, please. So before you submit your journal, definitely find the right journal, read the journal and read the instructions for authors and make sure that your manuscript complies. If you're not sure whether your manuscript is a good fit, you can actually write the editor and tell the editor, I'm thinking of submitting an article on so and so. Is this something your journal would want to consider? And editors actually like receiving that. There's a lot of work to get your article in the system, only to realize that it's out of scope or it's not a good fit for the journal. Run your manuscript by colleagues. Colleagues can give suggestions. They might know a journal that you don't know of, and they might be able to give advice for where you could submit things. Read the instructions for authors. I've said this before, and that's not too many times. You've got to make sure that your journal complies. Otherwise, it could be rejected before the editor sees it, and that's not what you want. Write a good cover letter. Tell us why this is important, why it's new, and why you think it's a good fit for the journal. If you do that, then the editor can have a conversation with you while you're not in the room and be able to, it helps the editor make a decision as to whether or not this article should be sent out for peer review. And then many journals will give you the opportunity to suggest reviewers. It's a good idea to do that. Something that many authors don't know is that very often the rate-limited step for the editorial team in terms of getting your article through peer review is finding suitable reviewers. Reviewers are volunteers. They're not paid. And so very often it's not easy to get 2 or 3 people to be able to review your article. So if you can recommend people who have the right expertise and might be available, sometimes that can help. Once you've submitted your article, the editor will either decide to send it to peer review or review it editorially and give you a decision. Typically that won't be a positive decision if it's editorial. But they could give you advice in terms of, oh, you might want to send this somewhere else, or you might want to fix A, B, and C and then send it back to us. If the editorial decision is negative, very often you'll get feedback that will allow you to improve the article and then be able to submit it either to the same journal or to somewhere else. But if your article does go through peer review, then you will get, in addition to editor comments, you get comments from reviewers. And unless all reviewers and the editors say, this is wonderful, we want to publish this now, that does happen, but it's extremely rare. I don't think I've ever been able to do that since I've been editor of AJLM. It's more common for the editor to say, oh, this is interesting. We like it, the reviewers like it, but A, B, and C need to be fixed. A, B need to be fixed. A, B, and C may be very small things, or they may be very large things. Okay, it's very important for you to read the instructions to authors again and also read those comments very, very carefully and then respond to them, use them to improve your article. And then you're going to prepare a revision that will be submitted back to the journal on the schedule that the journal typically asks for in order for you to be considered for publication. Try and respond to these comments. Typically, you send in your article with a rebuttal, and the rebuttal will be a point-by-point response to all of those comments. In each case, restate the comments. The reviewer stated that data was not robust enough, and so we have done additional experiments. Or the reviewer stated that this has been published before by so and so, but in fact the reviewer was in error. It's important to restate the reviewer's comments so the editor knows that you understood those comments. And then respond to them. You don't have to accept everything the reviewers say. They may be right, but remember they're peers, they could be wrong. But you do have to respond to all of those comments and explain how this has been used to review, to improve your article. Next slide, please. I do want to mention that sometimes the peer review process can be a little painful. Not all reviewers are polite. Sometimes even if they are polite, the comments are not easy to stomach. It's important to understand that peer review is there to improve your article, even if your article does not get accepted. Okay, you can get feedback that is useful. Sometimes you're right, but what you wrote was not clear enough. So if the reviewers are not understanding it, chances are the readers wouldn't understand it too. And you might have to rephrase something that was written in your article. And so if you're invited to submit a revision, don't use it as an opportunity to quarrel with the reviewer. Instead, use it as an opportunity to present a revised version of your article that is actually better. Okay, so I'm going to stop here with this presentation because I do want us to have quite a few minutes for a discussion with the panel. So I'm going to ask tech support to take the slides away. I'm going to ask all our speakers to join us on the panel. And if you have submitted questions to the Q&A of the platform, those questions are now visible in live. I've actually made all the questions live, and people can vote them up so that I can ask them as they appear. If you have questions that come to mind now, please can you add them in so that I can pose them to the reviewers. If there are not enough questions, I have many of my own. So we're going to have a vital discussion either way. And just to say, for those of you who are pre-registered and are on the Slack, you have the opportunity to ask further questions. So if I do see many questions coming in from somebody who is pre-registered, I may privilege somebody who's not, because this is their only opportunity to have their question answered. So our speakers are all here. We have Jenny Punt, we have Elshama Moko, we have Bethany Rammer, and we have Marie Chataway. And just to say that Ike Anya was unable to rejoin. He had a number of technical difficulties. But if you have questions that should be directed to him, any of us will be happy to try for answering them. So thanks, everybody. Those were some really great talks. I learned something myself. I always do. Thank you so much. And our first question is from Stanley Nurenda. And it's an obvious Jenny question. He's asking, and by the way, it had 4 upvotes, so lots of people are interested in this. Is it fine to have both your hypothesis and research question at the same time? Can you do too much of stating your purpose in your manuscript?
It's a good question. I think that you've probably noticed that, what do I call it? Productive repetition. And throughout a paper is sometimes more important than you think. I think that a question hypothesis are distinct. So they're distinct enough and should be distinct enough that you could have them back to back. And I do think that, as all of the speakers have shown, that you can repeat things, not in a way that's really boring, but a way that can help keep your reader centered. And I think your reader appreciates that. And I'll stop there, and I'm not sure I answered exactly the question, but I do think that a question hypothesis should come as a pair and can be repeated. Yeah.
Okay, thanks so much, Jenny. And I'll encourage you, as you're reading papers, look at the last paragraph of the introduction. Then you'll find these things. And when you've read many of them, then you have a sense of how you might be able to state your own hypothesis and questions. And for those of you on the Slack, one of the exercises is actually for you to do this on your own work. And we encourage you to actually use your own project rather than some hypothetical thing, because you can get actual feedback that you might be able to use for your hypothesis. So I have a question from Julie Okonji. This is something that I'm only mildly familiar with myself. So anybody can raise a hand to answer this. So what is a forecasting statement when you're writing a manuscript? Okay, I saw Jenny wave her hand. You want to go for it?
No, I think this was Elshama.
Elshama. Okay, but the question came before Elshama spoke. Elshama, would you like to answer that? Or should I?
It's forecasting is more or less like foretelling what is in your manuscript. So you've stated your hypothesis, you've stated your aim. Can you just give us a tidbit of what to expect in your manuscript? And it's usually very short, but at least it tells the reader, what do I expect? In the case control, it was the participants were female. It was in this setting, just very more or less like a result, but in brief. And sometimes it could also be your the result. Will I say, yeah, like a result? For example, the example I put there, where they ended up saying the laboratories now perform quality ELISA spot assays. So that's like a tidbit of what to expect in your manuscript.
Okay, thank you so much, Elshama. I have a second question from Stanley here, and he's asking now about the method section. Marie, maybe I'll ask you to answer this. So he's saying, is it okay to describe the method as described by the manufacturer's protocol, and then just put a citation? Is it okay to do that in a manuscript? Marie, you may have to unmute. Okay, we may have lost Marie, I'm not sure. I'll just give a couple of minutes and see if she can get her camera and microphone back on. Okay. Still can't hear.
It says I'm unmuted.
Yeah,
You
I can
can,
hear you now.
yeah.
Yeah.
So it was unmuted. I don't know why it wasn't. I think I was muted above. Yeah, so I was saying it depends how complex the instructions are for manufacturers. And going back to what Aruka says about read papers and see examples of how it's been written. Traditionally, we say according to manufacturer's instructions, and then we may write a brief summary. If you have made modifications to the manufacturer's instructions, then you definitely need to specify that. And sometimes if you are referring to previously published methods, if they've already been described, you can say as previously described and reference that. But if you do, like I say, read sort of different papers, depending on the method you're using, it can give you an idea of the best way to present that data. But I wouldn't say as described in manufacturer instructions and then start writing out everything that's within a instruction. We should summarize it.
Thanks so much, Marie. I should also let you know, if you don't already, that there are some, these are newer genres of papers that are really great. You can actually, if you've developed a protocol, you can actually publish your protocol as a paper. AJLM doesn't yet take these. It's something that I suppose we need to think about. But there's certainly journals that you can submit your protocol to and provide as much detail as possible. And also there are some journals that will allow you to provide detailed materials and methods as an appendix, as a supplementary data. I really encourage open methods. It can be so frustrating when you pick up a paper and you want to repeat what they've done, and then you just can't find, some small detail to be able to do that. Okay, we have a question that I need to question from Abdul Basit Opeyemi Abdurrahman, and he's asking whether. It's a bit difficult for me to understand the question, but let me see if I can figure it out. If there's no research in that area, he's asking whether it's acceptable to work on that. Maybe I'm not understanding the question fully, but it's, can I work on something that is new? The answer is absolutely. This is what the literature actually is for. But, a while ago, I was looking for this morning, and it's no longer online. There used to be a website where you could read the history of all the papers that Nobel laureates have published and have been rejected. Okay? So the newer your finding is, the more esoteric your finding is, the more careful people are going to be about accepting it. Okay, and this has its merits. It makes sure that a lot of rubbish doesn't get in the literature. And of course, it has its demerits when you're right. It makes it harder for you to publish. But then it places the burden on you to get more evidence to be able to support your new finding, which is really what we want. And this is the purpose of peer review. One analogy I've seen is actually in a published paper about writing. I just can't remember which one. That said, if I say I have a goat in my back garden, chances are someone's just going to say, oh, that's interesting, or peep over the fence and see, is there a goat there, or are there signs that my grass has been eaten by a goat? But if I say I have a unicorn in my back garden, then you're going to want a whole lot more evidence. Even if you peep there and you see something that looks like a horse with a horn, you're going to want to say, can that horn come off? Okay, and so this is how the scientific literature works. I'm hoping that's the question. It was difficult to figure out. If not, and if you're a participant that's registered, let's pursue it on Slack. And if anybody else wants to pitch in on my answer there, please do.
I think in that situation, I think it becomes difficult, especially for new authors, to sort of write the introduction, because they kind of want to say there's nothing, no one's ever done this before, there isn't anything to say about it. And I don't know how you really address that. Iruko or Jenny or Marie, you might have a good way to, I think you have to give the background in that case, and really set up your gap.
I think there's never nothing. There's always a foundation that you can bring to the paper. I think everything Aruka says is right on, and it is a challenge, but it might be worth it.
And sometimes we sort of write a bit of a caveat that this data, as far as it hasn't been published, because sometimes people may work on things, but if it's not published, it's not out there. So sometimes when you write in your discussion, you know, find this is the first report that we have found published, so to speak, rather than this is the first time it's ever happened, especially if you've got conferences and abstracts or things like that. So that can sometimes be worth putting that caveat in when describing something quite novel.
Thanks. Those are all really good ideas. And it happens all the time. I mean, early 2020, a group published a brand new coronavirus was killing people in Wuhan. So there's always new stuff. You just have to think about how to frame it. And as Jenny said, I think everything has a foundation. You know, there were viruses before, even there were coronaviruses before. And so those are things to think about. A question just came in. It's anonymous, and I think I know why. It says, what do you do when you know a co-author who should not be on a paper but is added due to rank? Now, this is what we refer to sometimes as guest authorship, you know, or gift authorship, which is worse. You're putting someone on there just because, you know, you feel if you don't put them on there, you'll be in trouble. It's really hard.
It does happen, and how you approach it depends on the institution you're in. You know you should confront it, but there are not always mechanisms to do that. But you should become familiar of those mechanisms, because it's a very difficult thing to confront. Authorship issues arise often. And I think your speakers here have done a really good job explaining what makes an author, but guest authorship and gift authorship does happen. I think all of us have experienced it, and I do think it behooves you to learn what your institution allows you to do in that case. You should be brave and you should be forthcoming, but if your institution doesn't really have mechanisms, it's very hard. So there's some sympathy. I'd love to hear from my colleagues.
Yeah, it happens all over, and I would like to say it will never happen again, but we know that it does. And also it is difficult, particularly if it's someone that's senior to yourself, and sometimes that's the culture and you might not be in a position to challenge it. Sometimes I do an approach of specifying at the end of the paper about the author contribution, to say we need to justify the authors, we can have acknowledgements, but they have sort of to contribute to that, and I get people to add, you know, contributions or suggest contributions. So sometimes that can help without being like challenging as such. I had it recently where I'm just writing something now to do with, it's only a quick letter of an emergent outbreak, and there's a couple of people from the lab, and I've contacted another section and they want to put like 3 people on it, and it's like, but it's difficult. But so I put that contributions at the end, and it's kind of made them additionally contribute to have justification for that authorship. So it's made them think about it, basically. So sometimes that can be a bit helpful. Like Aruka says, to start these conversations at the beginning, but like the anonymous person said, quite often someone will randomly pop in at the end. My most frustrating thing is when the only thing they comment in is how to put their title on what institution they're from. So yeah, it's an ongoing issue, but I think if we try to justify the contributions, sometimes that can at least prevent a lot of people adding guest authorships on there.
Thanks, Jenny. Thanks, Marie. That was really helpful. And I think one thing I would suggest is that if you have a choice of the journal to send your article to, try and pick one that actually has an author contribution requirement, because then you can write that in. And to be fair, I mean, we editors are supposed to look at those lists and query them. We don't do this as often as we should, but definitely writing it down will make it more obvious even to the person that maybe they're not an author. If they can't appear on the author contribution list, that is a hint. So we're coming to the end of the workshop, unfortunately, so I'm going to take one more question, and then after that I'm going to wrap up. As I mentioned before, if you're pre-registered, you can continue to question us on the Slack. So the last question is from Musa Ismail, and he's asking, is it okay if you've sent an article to a journal to email after a while and say, you know, I sent this in for a long time ago, and issues are being released without my paper. What happened? Absolutely. At AJLM in particular, we get a lot of these, and that's because our peer review is unfortunately slow. As I mentioned earlier, getting reviewers is sometimes a bit of a challenge, and unless we have at least 2 reviews, we really can't move forward. But sending an author inquiry, and the editor can tell you that, okay, yeah, we are working on this, it's in peer review. Or they can tell you that it's in peer review and it's stuck. We can't find any reviewers. And then you can choose, if you want to withdraw it and send it somewhere else. But if you want to know what's happened to your paper, you should definitely ask. If you have to ask several times, so be it. And also, I should say, as you get more experienced, consider reviewing, because this is a huge a roadblock in the peer review process. We have more people who are writing than are reviewing. And if you contribute to peer review, you can actually help speed things along. So I'm going to end now. I would have liked to ask a few questions of my own, but I can meet these people later on and do that, even on Slack. Before we go, I really wanted to say a few thank yous. Obviously to our speakers, Ike Anya, who you heard for a few minutes, Jenny Punt, Bethany Rammer, Marie Chataway, and Elshama Moko. You heard what they had to say both in this panel and in their talks. Really grateful for that. I'm also grateful to the additional mentors that we have on Slack, Akasin Odi and Pascal Ondua, who will be with us until Thursday. And then I want to thank the African Society for Lab Medicine that's organizing this conference, because as you know, satellite sessions are actually sponsored, and in this case the Society sponsored this session, so all of you could attend it without having to pay additional registration fees, so we're really grateful for that. And a number of us are members of the Council for Science Editors and get resources from there at no charge to us, which we can pass some of the wisdom on to you. So I really want to thank the Society for being a mentorship block for those of us who edit. And thank you all participants for joining us for this. We know we had several of you just drop in. We really thank you, particularly for the list of questions that came in. We hope you're able to take something away that was useful. And also thank you to those who took the trouble to apply and who will be discussing writing a bit more on the Slack. It's been a real pleasure for the journal to be able to host this workshop again, even in virtual format. And we hope that you'll get writing. We look forward to reading what you have to submit to our journal or to other places. Thank you so much. And enjoy the rest of the conference.
Bye.
Bye.